The human genome provides the instructions for life, but the operational reality of a human cell vastly exceeds those initial genes. The physical work is carried out downstream by structural hardware, ranging from stiff, rigid chains of carbohydrates to dynamic, interlocking globular proteins. In biological systems, function is an emergent property of three-dimensional stereochemistry. A molecule's physical form dictates exactly what it can and cannot do. We are going to trace the rules of this architecture. We will start at the atomic level, examining the rigid rules of chiral centers, and scale up to the thermodynamic folding of complex polymers. By tracking how simple monomers assemble into functional machinery, we can see exactly how these distinct structures converge in the metabolic core of the cell. Understanding the precise three-dimensional blueprint of a molecule is the only way to explain its behavior in human health and its failure in disease. Monosaccharides are the simplest units of carbohydrates. They are defined by their carbon chain length and the position of their carbonyl group. An aldehyde functional group makes them an aldose, while a ketone makes them a ketose. Because the carbon centers of these sugars attach to multiple hydroxyl groups, they are chiral. To standardize their spatial configuration, chemists use the D and L notation system, which arbitrarily uses the structure of glyceraldehyde as its universal reference point. This diagram illustrates epimers, saccharides differing at exactly one asymmetric carbon center. Here, a single inverted bond distinguishes mannose and galactose from glucose. D-glucose and D-mannose are C2 epimers, differing at the second carbon. D-glucose and D-galactose are C4 epimers, diverging at the fourth carbon. While drawn as straight lines on paper, these open chain structures exist in a constant dynamic equilibrium with closed cyclic forms. Cyclization occurs when the carbonyl group reacts with a hydroxyl group further down the chain, folding the molecule into a hemiacetyl or hemiketal ring. We project this three-dimensional perspective using a Haworth projection, numbering the carbons 1 through 6. This ring closure creates a brand new chiral center at the first position, designated as the anomeric carbon. The orientation of the hydroxyl group attached to this anomeric carbon dictates the molecule's form. If the hydroxyl group sits in the axial plane, it produces the alpha anomer. If it rests in the equatorial plane, it produces the beta anomer. This image displays the Haworth projections of glucopyranose. In an aqueous solution, the alpha and beta anomers are not locked in place. They undergo a dynamic interconversion process called mutarotation. The ring continually opens into its linear intermediate and snaps back shut, shifting between the alpha and beta positions. This constant shifting alters the specific optical rotation of the solution until it reaches a stabilized equilibrium. This chart shows that mutarotation is distinct from epimerization or isomerization. In basic aqueous solutions, heating causes sugars to isomerize through an enol intermediate, allowing, for example, D-fructose to convert entirely into D-glucose or D-mannose. The ability to shift between open chains, cyclic anomers, and varied stereoisomers give monosaccharides their biological utility. They act both as highly reactive metabolic fuels and highly specific structural building blocks. When monosaccharides polymerize, they link through a glycosidic bond. This forms via an acid-catalyzed reaction between the alcoholic group of one molecule and the anomeric carbon of another. During this reaction, the molecule forms a planar sp2 hybridized oxocarbenium ion intermediate. Because this intermediate is flat, the alcohol can attack from both faces, resulting in both alpha and beta glycosidic linkages. The nature of these repeated linkages determines the final polysaccharide. Polymers built from a single type of monomer are homoglycans, while those utilizing distinct repeating units are heteroglycans. The angle of the glycosidic bond entirely dictates the biological role of the molecule. We see this explicitly when comparing starch, a biological battery, to cellulose, a structural frame. Cellulose relies on linear beta-1,4 glycosidic linkages. This geometry places all hydroxyl groups in an equatorial arrangement, stabilizing into a rigid, insoluble chair conformation with immense tensile strength. Starch, conversely, uses alpha-1,4 and alpha-1,6 linkages. This creates a highly branched, accessible polymer specifically tailored for rapid enzymatic hydrolysis and energy storage. Mammals possess the alpha-amylase enzyme required to break down starch into glucose, but entirely lack the beta-glucosidase required to sever the bonds in cellulose. A geometric difference of a few atomic degrees renders one molecule food and the other wood. Scaling up, the sum total of all carbohydrate structures in the human body is called the glycome.
These complex sugar chains coat the surface of human cells, mediating everything from cell-to-cell adhesion to immune signaling. Sugars frequently exist as glycoconjugates, covalently binding to proteins and lipids to alter their function and guide them to their cellular targets. The macroscopic strength of a tree, the energy density of a potato, and the communication between our immune cells are all direct consequences of the microscopic angle of a single glycosidic bond. Proteins execute the actual labor in the cell. They are polypeptides, linear sequences constructed from a pool of 20 standard alpha amino acids, joined together by covalent peptide bonds. At a biological pH of 7.0, an amino acid exists as a zwitterion, with a positively charged amino group and a negatively charged carboxyl group. The molecule acts as both an acid and a base. What distinguishes the 20 amino acids from one another is the specific R group side chain attached to the central alpha carbon. This appendage determines the precise size, electrical charge, and water solubility of each unit. Protein architecture scales through 4 distinct levels of complexity. The primary structure, the exact linear sequence of the amino acids, acts as the fundamental thermodynamic blueprint for the whole assembly. This diagram of insulin reveals how the primary sequence is physically tethered together. Covalent disulfide linkages bridge specific residues, binding different sections of the linear chain into a stable shape. A protein naturally seeks its native conformation. This is the functionally active shape in 3-dimensional space that possesses the absolute lowest Gibbs free energy. This process is largely driven by the hydrophobic effect. In an aqueous cellular environment, the protein folds inward, burying its nonpolar, water-tolerant R groups deep in its core to achieve a compact globular structure. This diagram outlines Christian Anfinson's experiment on ribonuclease A, which proved that all instructions for this folding process exist solely in the primary sequence. Anfinson started with the intact, active native state. He applied 2 chemical agents to the protein, urea, a chaotropic agent that disrupts the non-covalent interactions, and mercaptoethanol, which chemically reduces the disulfide bonds. Stripped of supports, the protein falls into an unfolded state, a random coil lacking catalytic activity. But when Anfinson removed the chemicals, the sequence spontaneously refolded. Driven by internal thermodynamics, it correctly reformed its disulfide crosslinks, returning to its active native state. The precision of this thermodynamic calculation is non-negotiable. When the cellular folding machinery fails, proteins misfold and aggregate, a process that directly causes neurodegenerative conditions like the dense beta-amyloid plaques seen in Alzheimer's disease. When folded correctly, the 3-dimensional geometry creates a highly specific pocket known as the active site. Here, the protein functions as an enzyme, binding exact substrates to accelerate chemical reactions by factors of millions. Without enzymes, standard cellular metabolism would take years to complete a single cycle. While the geometry of a sugar relies on the fixed angle of a rigid chemical bond, the geometry of a protein is the result of a dynamic thermodynamic calculation, translating a 1-dimensional genetic code into a 3-dimensional biological machine. We have examined the structural rules of 2 distinct chemical architectures. How do these fuel sources and molecular machines actually interface to sustain an organism? Their chemical pathways converge in the mitochondria, specifically at the citric acid cycle, the universal metabolic engine of the cell. This chart maps the flow of the citric acid cycle, or TCA cycle. It operates as an amphibolic pathway, meaning it executes both the catabolic breakdown of molecules for energy and the anabolic synthesis of new cellular precursors. When proteins are degraded, their amino acid carbon skeletons are sorted strictly by their structure. Leucine and lysine route directly into ketogenic pathways like acetyl-CoA, while other skeletons funnel into glucogenic entry points like pyruvate or oxaloacetate. To manage this complex flow of carbon, the cell relies on specific chemical cofactors, such as pyridoxal phosphate, which assists enzymes in catalyzing necessary transamination reactions. In the end, the system forms a perfect loop. The
The proteins, acting as specific enzymatic catalysts, execute the very reactions that break down the carbohydrate structures to extract energy and synthesize ATP. Life relies on this seamless biochemical integration. The specific 3D stereochemistry of sugars provides the fuel and the frame, while the precise thermodynamic folding of proteins builds the engine that runs it all.